Okay, so today we will be revising kingdom fungi, the entirety of kingdom fungi from the topic of veterinary microbiology, subject of veterinary microbiology. Okay, so let us start. Kingdom fungi, these are eukaryotic non-photosynthetic organisms which contain cell wall. Obviously they contain cell wall which is made up of chitin and polysaccharide. So their cell wall is made up of mostly chitin but sometimes includes polysaccharide also. Okay. Now, if we talk about reproduction of fungi, then reproduction of fungi, it is of two types, asexual, sexual, and in both asexual and sexual, both is by the production of spores. Okay. Now, for respiration of fungi, it is of three types: aerobic, facultative anaerobe, and obligate anaerobe. Obligate anaerobe, ruminal fungi. Example is ruminal fungi. It is the example of obligate anaerobe, ruminal fungi. Okay. Okay. Next is habitat. Habitat saprobes, majority are saprobes, then parasitic, then opportunistic infection, and mycotoxin producing. So these are the main examples of main habitats of fungi. So what are the habitats of fungi? Saprobes, that is saprophytic. Majority of them are saprophytic. Parasitic, that is in disease causing. Opportunistic infection, that is disease causing, and mycotoxin producing, that is again disease causing. Okay. Okay. Now few terms. Though these two terms are very important with relation to fungi. So anamorph and teleomorph. So anamorph, part of fungal life cycle when it is in its asexual lifespan. So anamorph means the part of fungal life cycle when the fungus is reproducing asexual. That is called anamorph. And teleomorph means the part of life cycle of fungus when it is reproducing sexually. So that is the meaning of these two terms. Okay. Now, deuteromycetes of fungi imperfecti contains imperfect fungi. That is those fungi whose sexual phase is not known. Okay. So this is the meaning of deuteromycetes of fungi imperfecti. Next is there are 7 phyla which are recognized in fungi, 3 of which are of veterinary importance. So Ascomycota, Basidiomycota, and Zygomycota are of veterinary importance. Okay. Next is morphological forms of fungi. So there are two morphological forms of fungi. Morphology means external feature, external structure. So morphological forms are two: mold and yeast. So mold temperature, incubation temperature is 25 degrees Celsius, incubation time is 1 week, and yeast incubation temperature 37 degrees Celsius, incubation time is 24 hours. We have to learn this. This is very important. These two parameters are very important, both of these. Now mechanism involved in fungal disease. So there are two mechanisms which are involved in fungal diseases. Three mechanisms. Sorry, mycosis, mycotoxicosis, and induction of hypersensitivity. So mycosis means the fungus itself invades the tissue. Mycotoxicosis means that the fungus produces toxin, and induction of hypersensitivity. So these are three methods by which the fungus cause diseases. Next is dermatophytes. So dermatophytes are septate fungi which invades the keratinized structures of animal's body. Okay, so these are septate fungi. That means the hyphae are septate. That means they contain septa, that is cross walls, and they invade the keratinized structures of animal body or human body. Why only keratinized structure? Because of the fact that they have an infectious spore that is called arthrospore, and these arthrospores are released by fragmentation of hyphae, and this can take place only in the keratinized structures. Okay, so this is the reason because of which dermatophyte can only affect the keratinized structures, that is skin, hair, claw, nails, all of these. This is an aerobic fungus and causes characteristic ringworms or circular skin lesions. So this is the example. Now dermatophytes are divided into three anamorphic genera. Anamorphic genera means the three genera which are included in the which are always present in the anamorphic phase. That is asexually reproducing phase. So that is Microsporum, Tragophyton, Epidermophyton. Okay. Now we'll learn the example of these two later in this chapter only. So causes dermatophytosis. Dermatophytes causes dermatophytosis, and dermatophytosis, it is a zoonotic disease. It is a zoonotic disease, that is it can be transferred from human to animal and animal to human, and human infection by Microsporum canis. So this is the human infection is caused by Microsporum canis. Okay. Now types of dermatophytes. So there are three types of dermatophytes: geophilic, zoophilic, anthropophilic. So if we talk about geophilic, then they inhabit and replicate in the soil. Zoophilic, they are obligate pathogen of animals, and anthropophilic, they are obligate pathogen of humans. Okay, so these are the basic meaning. Obligate here means that they cannot survive without animals, and these anthropophilic, they cannot survive without humans. Okay, now these are the examples: anamorphic genera, Microsporum, Tragophyton, and Microsporum canis, Gypsium nanum, and Bentagrophyte, Tragophyton, Mentagrophytes, and Tragophyton verrucosum. Okay. Next is yeast and disease production. Next we'll talk about yeast and disease production. So if we talk about yeast and disease production, so yeast are eukaryotic, unicellular, or round or oval in shape microbes, morphological form of yeast or fungi. Okay. So during asexual production, they produces bud, daughter cell, or blastoconidia. As we all know that yeast reproduce by budding. So those buds are called buds, daughter cell or blastogonidia. Their habitat is plants and on skin and mucous membrane as commensals of animals and humans. Yeast incubation period is 24 hours. Incubation temperature is 37 degrees Celsius, as we all know. Some yeast produce pseudo hyphae. Now, no yeast produces hyphae, but there are some yeast which produces pseudo hyphae. For example, Candida albicans. So Candida albicans is a yeast which produces pseudo hyphae. Pseudo hyphae, what is pseudo hyphae? So when these blastogonidia or buds are arranged in a linear fashion, that is, they are not separated from each other, they are arranged in a linear fashion and forms a long chain of buds, that is called pseudo hyphae. That is not true hyphae, but it resembles a hyphae. Okay. Now, opportunistic infection. Now, any infection which we can get from yeast can be divided into two types, exogenous as well as endogenous. So exogenous means when the fungus, then the yeast infection derived from the environment. Endogenous means when the infection is due to the overgrowth of commensals or microflora. Okay. Next is Candida species. So Candida species incubation temperature is 37 degrees Celsius on SDA. Obviously it is a yeast, so it will be incubation temperature will be 37 degrees Celsius. In animal tissues, Candida albicans exhibit polymorphism. Polymorphism means that poly means many, morphism means morphology. So many morphology. So there are three morphology: budding yeast cell, pseudo hyphae, and true separate hyphae. Budding yeast cell, pseudo hyphae, and true separate hyphae. Okay. Next is habitat. So habitat, these are commensals. That is a form, we can say, a form of symbiotic relationship where the organism lives without harming its host. Now more see, this Candida albicans is more a parasitic rather than a saprophytic fungi. Why? This is because of the fact that this fungi is mostly recovered from the animal tissues or human tissues. Okay, that is why. Now, germ tubes are formed when the cells of Candida albicans are incubated in serum at 37 degrees Celsius for 2 hours. So these germ tubes are the characteristic structure of Candida albicans. Okay. Now, pathogenesis and pathogenicity. So Candida albicans is the principal yeast which causes infection in animals, and it possesses a number of putative virulence factor. I don't know the meaning of putative. I myself don't know the meaning of putative. So that is the fact. And it can be observed in using India ink preparation. So this is also one of the points about Candida albicans. Okay. Next chapter is mycotoxin and mycotoxicosis. Within fungus only, we are studying fungus today. So, if we talk about mycotoxin, mycotoxicosis, and mycotoxin production is, so what is mycotoxins? What are mycotoxins? Mycotoxin, these are the secondary metabolites of fungal species when toxinic strains of fungi grow on crops, feed or pastures. Okay, very easy. Now, what is mycotoxicosis? Mycotoxicosis, acute or chronic intoxication following ingestion of contaminated plant or animal, but a plant material. This is not animal, this is material. Okay, so this is the meaning of mycotoxicosis. So mycotoxicosis is the infection, and mycotoxins are the chemicals or poisons. Mycotoxicosis are produced by fungus belonging to mainly Penicillium, Aspergillus, and Fusarium. So this is the this is an important point. And next is that it is required for, what are the conditions required for fungal growth? Availability of a suitable substrate, moisture, temperature, oxygen. These are the basic points. Now, what are the property of mycotoxins? So these are non-antigenic, that is, they do not lead to a production of antibody. They are of low molecular weight and they produce clinical signs related to central nervous system. Easy? Okay. Now, next chapter is aflatoxins, aflatoxicosis. So it is caused due to the ingestion of aflatoxin. And what are aflatoxin? These are a large group of chemicals produced by toxinic strains of Aspergillus flavus and Aspergillus parasiticus. So this point we have to learn. Aspergillus flavus and Aspergillus parasiticus toxinic strains produce aflatoxins. Okay. Now, types of aflatoxin. There are 4 types of aflatoxin: B1, B2, G1, G2. Pathogenesis, and animal ingested aflatoxin, it went to GIT, it is transported to liver, then aflatoxin are broken down into toxic products and non-toxic products. These toxic products causes toxicity. Okay. Next chapter is Aspergillus species. So Aspergillus species, it causes respiratory pathogen in animal, and mode of action opportunistic and mycotoxin production. So this is one of the tables, and this completes our fungus chapter.